Hello, my name is Fiona Brennan. I'm a soil microbiologist in Teagasc and Johnson Castle. And today I'm going to give a quick summary of some of the research we've been doing investigating whether microbial bioinoculants can be used as biofertilizers in grasslands. So why are we interested in grasslands? Firstly, grasslands constitute about 70% of all agricultural land. And in countries like Ireland, this can be up to about 90% of all agricultural land, making it a very important land use. All food systems need to be both economically and environmentally sustainable, leading towards a push towards reducing chemical fertilizer in these systems, which can be both costly for the farmer and also can be associated with negative environmental impacts on water quality and on air quality, including greenhouse gas emissions associated with fertilizer application. So finding alternative methods of providing nutrients to plants and supporting growth of plants can be very advantageous on multiple fronts. So biofertilization strains are one technology or one option that could be used to replace these fertilizers. And one of the groups that's often used for this purpose is arbuscular mycorrhiza fungi, or AMF for short. These are fungi that form symbiotic relationships with plants. And in return from carbon from the plants, they provide a range of nutrients to the plants derived from their extensive networks, mycelial networks below ground. In this project, we used 3 of these strains together. We also looked at another group of organisms, plant growth promoting rhizobacteria, and these were able to mineralize nutrients and improve plant uptake. So we looked at 2 bacterial strains, a bacillus strain and a Paraburcolderia strain. These isolates were used in tandem and also in singly. We looked at how these strains did in both monocultures of perennial ryegrass and also more diverse sward types where we incorporated legumes and herbs. Our main questions were, did these strains colonize? Do they persist long enough to have an impact? What's the impact on the quality and quantity of the herbage? And what's the impact of soil fertility in terms of whether these strains work or not? We're also interested in the impact on the wider plant and soil microbiome. Our key take-home messages were, yes, we could get successful colonization of these strains, and they did persist. We found in terms of yields, sward composition had the biggest effect on yield, but for some consortiums, so for example, our AMF applied in concert with the bacillus, we did get increased uptake of calcium, of nitrogen and phosphorus, and trends of increased yield. We found, or we learned, that compatibility really matters in the formulation of these consortia, and some organisms work much better together than others. Also, the soil fertility context is really important in terms of whether these organisms will work or not. So just finally, to acknowledge my colleagues in Teagasc, particularly Israel Ecoy, who did most of this work, and our partners in AIT, Christian Hansen and Enoch, and our funders for the Horizon 2020. Thank you very much.
My name is Renny Groben. I'm a project manager at the microbiology group at Matis, and I will give you a short presentation about the research on increasing bioavailability of sustainable aquafeeds and food safety. With the growing global population and the need for healthy, nutritious food, seafood is in demand as never before. However, current fishing practices put a lot of pressure on marine ecosystems, and fish stocks worldwide are often in danger of overfishing, and this makes this practice unsustainable in the long run. Aquaculture, which is the rearing of aquatic organisms in large tanks on land or in open pens in the ocean or in lakes, has the potential to solve this problem and produce high quality food in large amounts. As a result, aquaculture quickly became the fastest growing industry in the food sector. There are, though, problems associated with growing aquatic organisms in aquaculture, and one of them is the need for sustainable sourced food and feed. At the moment, the necessary protein sources and largest component of aquaculture feed is fish meal, meaning that low-value fish is caught in the ocean or reared and to feed the more valuable fish in the aquaculture. This system does, of course, not solve the problem of overfishing and depletion of fish stocks, and in the long run, this is also not sustainable. So we need to find alternatives. In MASTER, we did trials with different kinds of alternative protein sources in aquaculture feed for Arctic char and Atlantic salmon. We used proteins from waste streams of the dairy and beverage industry, and as well as insect meal, and replaced a certain percentage of the fish meal in the feed with it. In addition, we supplemented the feed with selected prebiotics and potentially probiotic bacterial strains to test if they increase the bioavailability of these feed. We think that these might change the composition of the microorganisms in the fish gut and have beneficial effects. We measured the impact of these treatments by analyzing the growth rates of the fish, the quality and sensory values of the fish fillet, the gut microbiomes, and the product safety through microbial and chemical analysis. The results showed that fish meal could be replaced by up to 15% with alternative protein sources under prebiotic supplementation without any negative effects. There was even an indication that some of the probiotic bacterial strains had growth-enhancing properties both for Atlantic salmon and Arctic char when they were administered with the feed. Furthermore, the study showed that these probiotics inhibited the growth of pathogenic Listeria on fish fillets without having any adverse effects on the quality of the fillet. So this makes them a valuable tool to improve food safety in the aquaculture. We think that these beneficial effects could have been achieved through modulation, meaning changes of the microbiome, the microbial composition in the fish gut. To summarize, our research has been shown to the possibility to use alternative and sustainably produced protein sources in aquaculture in combination with pre and probiotics. Not only have they no negative effects, but we could even show that they might increase growth and reduce infections with the pathogenic Listeria, making them a promising way forward towards a safe and sustainable aquaculture production. This work was done within the MASTER project by researchers from Mathis and Ifremer, and we would like to thank all the colleagues for their successful combination.
Oh, hello, everyone. I'm David Yañez, a researcher working at the Spanish Research Council in Granada, Spain. And I'm going to quickly tell you some of the results we obtained combining ruminant enteric methane inhibitors with hydrogen acceptors. We know that ruminants such as cattle, goats, and sheep, they establish a very strong relationship with a complex microbial ecosystem that degrades part of the plant material that come into the diet, and in that degradation activity, they produce different compounds, one of them being different volatile fatty acids, acetate, propionate, or butyrate, that represent the main source of energy for the ruminant. In that kind of fermentation, also hydrogen is produced, and methanogenic archaea in the rumen are responsible for using that hydrogen to produce methane, so the fermentation can continue. The production of this gas, methane, which is a potent greenhouse gas, contributes significantly to the emissions of greenhouse gas emissions of the animal agriculture. So different strategies have been developed, one of them being the use of methane inhibitors that inhibit the activity of methanogenic archaea to reduce the production of methane. When these methanogenic archaea inhibitors are used, there is also an excess of hydrogen produced in the rumen that is wasted because it's not used to produce energy yielding compounds. Different alternatives have been proposed in the last years to try to capture this excess of hydrogen, so the animal can not only reduce the production of methane, but also obtain a benefit from the metabolic point of view. Some of these initiatives include the use of acetogens, sulfate reducers, and nitrate reducers, but there are some limitations associated with these strategies. We are proposing a different strategy using different phenolic compounds that are present in plant materials, that when they are degraded in the rumen, they produce volatile fatty acids that can further contribute to the energy production for the animal. So to test this hypothesis, we ran a different set of in vitro trials combining a potent methane inhibitor like Asparagus taxiformis, a red algae, with a range of different phenolic compounds, as you can see here, to see what the effect was on rumen fermentation. And we could see that when asparagus was used, there was a significant reduction of methane production that was also associated with a huge increase in hydrogen production, as you can see here. When Asparagopsis was combined with these phenolic compounds, in some cases, this excess of hydrogen was reduced. And in the case of fluoroglucinol, one of the phenolic compounds, there was an increase of total volatile fatty acids. So meaning that this hydrogen in excess was used basically to produce compounds that the animal can use as a source of energy. And this is basically as a result of fluoroglucinol being degraded in the rumen to produce acetate, and in that process uses hydrogen, and in this case, part of the hydrogen that is generated in excess when methane is inhibited. We had a chance to further test and confirm this in vivo trials in goats in respirometry chambers, as you can see in here in this photo. Again, using a controlled diet compared to either Asparagus taxiformis, fluoroglucinol, or the combination of the methane inhibitor with the phenolic compound. And we could see again that Asparagus taxiformis significantly reduced methane. But when we combined the use of Asparagopsis with fluoroglucinol, there was not only a reduction of methane, but also an increase in total volatile fatty acids, confirming what we already obtained in vitro. So the conclusions are that combining a potent methane inhibitor such as Asparagus taxiformis with a phenolic compound that uses the excess of hydrogen represent an attractive combination for farmers to reduce methane emission, but also provide more energy for the animal. Thank you very much for your attention.
Hello, everybody. My name is Vincenzo Valentino. I am a postdoctoral researcher from the University of Naples Federico II, and the title of my presentation is Novel Strategies for Microbiome Mapping in the Food Industry. Basically, we know that in the food industry, microorganisms are present on surfaces during the food processing. However, even though we clean the surfaces, we know that a bunch of microorganisms are still present on these surfaces. Basically, the microbial load resisting on surfaces after the cleaning and disinfection is quite limited. So they are difficult to be studied using current methodologies. So we wondered, who is there, what can they do, and also why and how do they survive? To answer these questions, we applied the standard operating procedures developed within the master project, which are based on detection and study of the microbial DNA. Well, to answer this question, we test the procedures in two industrial settings. One produced the minimally processed vegetables, while another one produced ice cream. What we did was basically collecting environmental samples after the cleaning and disinfection, collection of ingredients and products from the first production following the environmental sampling, and then in the laboratory, processing of samples, detection of DNA, and data analysis according to the standard operating procedures developed. So here are the main outcomes from these studies. We identified the communities residing on clean surfaces, highlighting that they might be very complex, so made of lots of species. This is a very interesting result that was impossible to achieve using the current methodologies that are used to study the microorganisms associated with the food industry. Also, we detected microorganisms genes involved in production of biofilms. Biofilms are thin layers strongly attached to surfaces, embedding and protecting many microorganisms from disinfectants. We also, our results also are in accordance with the previous evidence, which suggests that the presence of these biofilms might also lead to selection of superbugs that are microorganisms that show antimicrobial resistance to several compounds. Finally, we also predicted that a great proportion of resistant microbes living and selected on clean surfaces might be transferred to the final product. And this might arise some problems regarding the quality of food products and also for safety reasons. So overall, the advantages of the standard operating procedures that we test and the future outcomes are here presented. Basically, we were able to quickly detect spoilage and even pathogenic bacteria. And so these procedures might be useful in the future to reduce food waste and also to prevent foodborne outbreaks. Also, we were able to achieve a higher resolution in profiling microbial communities and their potential compared with traditional methods. So overall, in the future, stakeholders might soon implement these standard operating procedures as a routine practice to monitor microbial communities residing in the industry. This may be very useful to develop ad hoc sanitation strategies that are tailored towards some specific microorganisms that need to be eradicated from the specific industry. So thank you for your time.
Hello, my name is Ines and I'm a PhD student at IPLA-CSIC, and today I'm going to talking about the development of probiotic ingredients from apple waste. The cider sector in Asturias produced around 12,000 tons of apple pomace per year that is used normally as a compost for animal feed or just waste. We have focused our work on apple varieties under PDO Cider of Asturias, and we selected 9 varieties based on their resistance and production. First, we made a general characterization with these 9 varieties, and we saw all of them has a high mineral content, high fiber, and galacturonic acid content. Because that, we made a pectin extraction with these 9 varieties. The raw material was freeze-dry, green, and low the pH with a citric acid at 2.5. Then we made a conventional citric acid extraction, and we obtained around 10% of the yield. With the results, we selected these 3 varieties, Solarina, Regona, and Perico, because they are interesting pectins due to their structural difference. With these 3 varieties selected, we made another 3 pectin extraction methods, the same conventional method but at a low temperature, and 2 more methods with ultrasound, bath, and probe modes. The yield was not improved, but we obtained pectins with different structural characteristics that can improve the fermentative properties. Based on these results, and with these 3 varieties selected, we made a batch fecal fermentation. First, we made a simulated digestion with all these substrates, and then fecal fermentation with fecal samples from IBD and healthy donors. We made this fecal fermentation with medium and 0.5% of substrates, and we take samples at different times for taxonomy sequencing. The results of this part, we saw a statistical significant increases in abundance at the genus level. For example, Akkermansia, that is associated with intestinal health, and Turicibacter and Dialister, that are anti-inflammatory microorganisms. Also, we saw the relationship between microorganisms are different depending on the type of the donor and substrate being studied. As a conclusion, the structural variety of pectin and apple pomace influence the fermentation potential and the ability to modulate the intestinal microbiota. These results open the way to achieve its valorization through the formulation of new probiotics for particular population groups, and the use of both pectin and apple pomace favor the representativeness of some microbial population that decrease in IBD. Finally, I want to thank everyone who has made this work possible.
My name is Coral Barcenilla. I'm part of the University Lyon Partner, and we've been involved in one of the work packages of the master project related to the design and validation of biopreservation strategies for meat products based on the use of Lactidase bacteria. Foodborne outbreaks caused by the consumption of contaminated meat and meat products still occur with important negative effects on the public health. Listeria and Salmonella might sound familiar, as they are two groups of bacteria that can originate zoonosis associated with the consumption of meat products. For example, Listeria monocytogenes is a cause of major concern, as it has the highest fatality rate in Europe among the rest of bacteria. This, together with the increasing rejection of consumers against chemical additives or the demand of green label alternatives, has promoted the development of new strategies for the preservation of meat products. The use of beneficial bacteria, such as Lactidase bacteria, or the products that they can produce, can be used as natural antimicrobials to compete with harmful or spoilage microorganisms, and so extend the shelf life of meat products. Our research group has studied the viability of applying Lactidase bacteria in different meat products to control the growth of those pathogens. We've tried different products, different conditions, and different types of applications. For example, we have applied the bacteria on the surface of the sliced food ham, or mixed with the ingredients to elaborate a fermented sausage, or in marinades where pieces of pork were dipped in. To evaluate the effects of this application of Lactidase bacteria on the products, we have evaluated different parameters such as the bacteria present, the color and texture parameters, if any important acidification occurred in the products, and also a sensory panel evaluated the final product. So the results that we obtained are shown here, some of them. We can see the growth of Listeria and Salmonella during the ripening period of a fermented sausage, and we can see that on the last day of the ripening, while there's still high counts of both pathogens, as we can see with the darker lines, when the Lactidase bacteria were applied in the product, we can see that those pathogens were reduced, as we can see with the light lines. In addition, other bacteria that can cause the spoilage of food products can be controlled with the use of these Lactidase bacteria. Procotrystium specta, for example, is a well-known spoilage microorganism that grew spontaneously on two experiments performed in a fermented sausage and in cooked ham. But when the Lactidase bacteria were applied into these products, there was a reduction of these spoilage microorganisms. So, as a conclusion, we can highlight that the tested Lactidase bacteria demonstrated promising results to fight against different foodborne pathogens, that they were effective in different meat products and with various packaging conditions, and that the bacteria applied did not have any detrimental negative effect on the meat product quality. If you have any questions, we'll be happy to answer them.
Good afternoon, everyone. I'm Nicolò Carlino, PhD student from the Segata Lab of the University of Trento, and today I will present you our project entitled Curated Food Metagenomic Data, which is a large database of food microbial communities. But why do we need such a database? So we have known since many years that microbes have an important role in food, which can be positive, such in case of fermentation, or negative in case of sporage or pathogens. And up to a few years ago, it has been studied through classical microbiology methods, such as isolation, which had strong limits, for example, the difficulty or impossibility to cultivate microbes, and also the limitation to study one microbe at a time. Almost a decade ago, we had that technical revolution, which now allows us to study the metagenomes, which are the disordered DNA information of the whole microbial community. Meaning we have a lot of data, but in order to take out sense of it, we have to use a computational pipeline and a reference database. And that's what we did. So we built the first database of food metagenomes, CFMD, which is large and comprehensive, including over 2,500 samples from all over the world, almost 2,000 of which coming from the master partners. We have samples from more than 50 countries, 5 continents, and from very various food sources. And the master contribution was fundamental not only in terms of quantity of samples, but also in the increase of quality, that is to say, the information per samples, and also from food types, since we are now able to study foods that weren't available before. And that's our result. We have been able to reconstruct almost 300 new putative novel species, over a total of 3,600 species different found in food, among which we have been able to characterize them among those which were commonly shared and those that were type specific. But all in all, the bottom line is that communities are very food characteristic. So our result is the construction of a public database with the unprecedented contribution of Master, whose main results is the demonstration that food microbial communities are very diverse and still in expansion, since we are discovering new species every day working on it. And these have very important impacts, both in the scientific community, as this can be used as reference, but also in the industry, since it's exploitable for food safety, quality control, and also the production optimization. But last but not least, you can also use to identify probiotic species and prebiotic food. So thanks for your attention, and thanks to all our collaborators.